Segundo. Vamos a su... Okay. Hello. Good afternoon. So we are going to start this session. The first speaker is Eduardo Colombo. Eduardo Colombo has done a PhD in physics at the Pontifical University of Rio de Janeiro in Brazil, with a sandwich period at the IFISC, the Institute of Complex Systems in Palma de Mallorca, Spain, then a PhD in Princeton, and now he is at the Center for Advanced Systems Understanding, that is a Helmholtz-Zentrum in Dresden, Germany. So he's going to talk us about pulsed interaction as a route to self-organization. Can you share the screen?
I don't see how to.
Sorry, try again, please, to share your screen.
Oh, yeah.
The other, I mean, the people who are here cannot be seen. Where should I see them?
Can you? Yeah, I think you can see my screen. Yeah.
Great.
Yeah. Do you see it in full screen or?
Yes.
Yes. Okay. Okay. Thank you very much for the invitation. I really appreciate. I'd like to thank Albert for setting up this space and other members of the consortium, in particular Celia, who invited me to give this talk. Celia was my advisor during the PhD, and now a colleague for many years. So I'm really happy to present this. It's nice that the audience are physicists. Typically, I present this work in some interdisciplinary context. So yeah, I think you're going to appreciate the results from the mathematical perspective. So this work touches a little bit the foundations on how pattern formation occur. So we know this from Turing works, and usually you have some sort of a first layer, which is your population, sometimes referred as an activator, and you have a second layer where an inhibitor or a mediator of interaction travels and creates a feedback loop between the agents in the population. So to establish these two layers of interaction, you have a reaction diffusion also occurring as they move through space. And this communication, there are many pathways for it. So you can have cells releasing some chemicals and the others are receiving it. So it basically, by releasing and absorbing a certain substance, you can establish this type of communication between cells. Also, you can have many different types of interactions. You can have root interaction in vegetations, which makes the interaction non-local because you have these structures that grow underneath the soil and actually mediate the water competition. So you have the vegetation and the water. The vegetation would be the population. Water is the mediating substance there. You have individuals using calls. They emit some signals in terms of sound, and the others perceive it, and this can be very complicated or very simple. So in general, what it's done, you create a set of equations for the population and for the many or one mediator. They interact, they have a reaction term. Not sure if you see my cursor, but you have the F and G's are the functions that are the reaction rates. And you have the diffusion operator here in finite difference. So you have this set of equations and you want to know when patterns can occur or not. So this is important for basically the emergence of spatial structures, which will have consequences for, let's say, developmental biology, ecology, many fields beyond physics. So you have that system, you do some sort of linear stability analysis, you go to the homogeneous state, and you ask, oh, is this homogeneous state stable under perturbations? And then you get the eigenvalues as a function of the modes, and you want to know if there is any that goes positive. Yeah, you want to know if there is instability of a given mode. This has been done for models. Some are very classical and well studied. This is one of them. You have an activator and an inhibitor, and they interact and they create these nice patterns. This would be the concentration of the activator population. You need some conditions for it to occur. I'm not going to do the math for you. This is 1972. 2001, I studied this during my PhD. You have this was a PRL. It was a really strange to me as a student to see like vegetation patterns in PRL at the time. Say, okay, we can do that. And then you have some sort of reaction diffusion as well, and you have the density of biomass, the vegetation, and W is the water, and you have these two layers of things interacting and creating these spatial patterns. And there are many, right? There are many across scales. They appear. You can have patterns at muscles. Some very nice work being done on muscles. You have these new results on sacked cornea. You see these labyrinths. Here you have fish skins and bacteria in petri dishes, and vegetation across many environmental conditions. So you have these many patterns in nature. But my point and the point of the work we did is that when you perform this type of equations, there is some sort of smoothness to the dynamics, right? Things are interacting equivalent or proportional comparable rates, better say. But you see that, for instance, if you start to analyze other types of communications, this is a case of fireflies. And of course, the interaction between them are not a continuous process, but they are like flashing in time. And you can see some illustration here on the left by this the lab by Urit, which is like doing very groundbreaking work in firefly communication. And they, of course, they not interact. They are not continuous interact, as in the case of vegetation, where water is continually diffusing and being absorbed by the roots. The interaction is very flashy. And of course, in the case of calls as well, right? You have these animals shouting or doing some weird sounds, and they hear. There is actually some sort of communication and codification here as well, and they actually overlap. They avoid overlapping, so they are smartly using the sound space, because the interaction is post and not continuous. So you leave space for signals to be emitted and clearly identified without the interference of others. So in general, you have the whole space of possibilities. You can have fast signals, but that propagate fast across space. You can have everything. So let's take one example here, scent marks. So when a wolf crosses a tree, it leaves the scent mark there, and this will last for perhaps a day, but this is only perceived locally, right? No other individual that's not at the location of the mark will feel it. Okay, a little bit around, but need to be close to it. And you have, for instance, calls, which is in the other extreme, where it lasts for a second, but it can reach us very far away. So you have communications occurring in these different aspects or characters. And one of the important things that occurs that when you become pushed, when interaction become pushed, it starts to not overlap. And this is, as you may appreciate this is going to be a consequence to, let's say, the superposition principle when you solve, let's say, when you try to do like some linear stability analysis, and in the linear regime, you can just basically sum the contributions of the pulses, let's say, that this thing. And if it's linear, you can do that. But and it doesn't matter if they are overlapping or not, right? If the dynamics is linear, so it doesn't matter for you. But if the signals are propagating in a non-linear way, the superposition principle are going to be put at risk in this case, right? Because you're going to have different solutions when those pulses are overlapping or non-overlapping. And there are many interesting ecological questions that you can think about it. So let's start with a very minimal example. This is the first system we explored. So you have a population of entities that are replicating themselves. They move at random, and they involuntarily, so they are emitting a signal that's not conditioned to anything. There's a rate, and these signals basically harm the others around. So it's by like a defense mechanism or to put others away, you just keep sending bad stuff like toxic signals, so they don't approach you, or they die before approach you in this case. And then the main difference between what you would see in a simple Turing classical type of equation is that this guy here, which is the influence of the population in the field, that is when the individual is releasing the signal, so it's the individual is promoting the propagation of the signal. This is continuous on those equations I showed you before. But here is a post. So sometimes this is like a noise that's density dependent. It's conditioned to the density of O. So you have this type of equation, and then you can study two limits. You can set the limit when the signal propagation is fast, so you're going to have the flash, and you have a quick propagation and a quick dissipation, and when the signal is very slow. And you can solve these two limits in general, and for a specific case, you can do a little bit more. So what you do, you take in this low limit, you basically the noise averages out, right? Because the signal is like is perturbing the signal field too fast for the response. So basically, this rapid fluctuations are averaged out, and then you recover the classical type of equation you're looking for. So you have this average dynamics because the signal is too fast for the response. And in the fast limit, you have that when the population perturb the signal field, immediately like a profile appears, right, really fast. So you can see in this plot x, the space, and time is passing. So the signal spreads and rapidly stays in a profile. So this will be the light of the firefight. So the light comes, stays a little bit, and then dissipates. The fireflies is not a good example here because the light of the fireflies don't kill the others, right? But it's just an illustration. So you do it again, but when you do that, you arrive to a different equation, which is a single equation, not two. You have a single equation for the population, and it's non-local. So when you change, you are keeping the dynamics the same, right? The operators that describe the reaction rates and the movement are kept the same. So this fancy L and the other L, they are kept the same when I'm changing only this pulsed or not pulsed smooth type of interaction. So this is what we try to argue, like a detour from what you would expect in a Turing-like system, because you have the same dynamics. So you have distinct stability criteria when this happens. So, and yeah, there are many mathematical ways to see that. One is that when you look for that first curve, I'm going to go back, sorry. So this first curve I showed you here, which is the growth rate of the mode as a function of the wave number, right? For the slow case, when you fall into the Turing-like thing that you see on the left, you have like a second, fourth order polynomial, right? In K4, K2, or I think I'm right. But when you have a non-local equation, this stability criteria is going to be an infinite series, right? Because this guy is going to be an infinite sum of k's at different powers. You can show that. I'm not going to go into details, but there is some nice mathematical ways to see this happening. Yeah. To show you a concrete case, we used nonlinear dynamics for the signal because of what I anticipated before, because there's involved this superposition principle and the breakdown of it. So I'm going to use this nonlinear decay diffusion equation. So you have a nonlinear decay rate and a nonlinear diffusion coefficient. This equation has been studied in different forms by many that you might know, Plastino, Tissalis, Buchner, but of course also by myself and Celia in these two papers. So they have biological motivations, and it has biological motivations behind it. So what we did was basically to show when we go from for this model, and assuming this population dynamics, basically diffusion and growth, we see this transition when we go from is moved to boost. So we see that the amplitude, or first that there exists a dominant wave number, right? If you take the Fourier transform, you see a clear peak, and that the size of the peak is relatively big. So this is what's showing here. I think the C, the white circles is this coherence, is the size of the peak of the dominant mode relative to the background node, and you see the patterns emerging only when the dynamics is pushed, and remember, with keeping the same dynamics. So this is what you see. So you see in this low signal, you see pulses everywhere, things are being agitated, things die, grow, but there is no clear coherence. But when you see the fast signal case, when you have flashy interactions, you see the formation of patterns. We extended these results so before this case was about I send a signal, I cue you. So this is the other example now is I send a signal, this signal make you move away. It's like a repulsion force. And as a repulsion force, again, mediated by this propagation of this mediator. So with the same result, we knew this more or less already, that we're going to see the same result, that when you have this move, when the signals are fast or slow, we're going to see a critical transition between how the population is organized. So I think I have maybe 5 minutes. So this is some theoretical work. Some of the criticism we received about this work is basically we found it very hard to give an actual biological example where a system would make this transition from slow to fast. We speculated a couple of cases, but we couldn't provide like like a sharp example. So this was like some frustration I had, and I've been looking through like data and collaborators that provide scenarios that I could find this type of signals of interactions. So I've been working now in my current institute with the PI is Justin Calabrese, so he has done a lot in methods for ecology, and we've been studying, for instance, these two animals. You have two animals, the Segre and the Ziggy. You have them here, and they sometimes they meet in this overlap of the probability distributions of their location probability, right? So, and when they meet, something happens, and we've been trying to quantify this interaction, which is hard in the wild. People have done in the petri dish or in a lab scale experiments, but in the wild it's really hard because data is not so high frequency as you would do in the lab. Yeah, we have many issues, but we've been trying to apply this pulsed or not pulsed interactions on how this home or the probability distribution of the individual is modified. And we've been working on a theory for this in collaboration with Luciano De Favre and many colleagues. And the importance, just to finish, I know that I might be ending my time, so just to finish, why this type of understanding, like fundamental understanding, is important. It's because this opens a very powerful connection between scales. So macroscopic things we can observe. We can observe these patterns I showed you. We can clearly observe from satellite images, as you can see here, the beautiful patterns formed by phytoplankton. These things are easy to understand, and if you know that some qualitative difference is associated to how things are behaving at small scales, here basically zooplankton interacting with the phytoplankton in a complicated way, or here if water or there is another type of mediator playing a role, which is a very constant type of discussion in vegetation patterns. So it opens like practical applications and actually bring a lot of ecological value to the theory. Finally, just to thank everyone. This work have been done with collaboration with Emilio that you see here, with Cristobal. This started many years ago, and then we slowly managed to accomplish that in a recent paper that we extended the framework. Many of the results were also studied by me with Celia and Luciano more recently, and Justin Calabrese and Ricardo Martinez Garcia are now very close collaborators on this interaction with ecology and using data to understand interaction between animals. So yeah, I think this is all I prepared for today. Thank you very much for this opportunity.
Thank you very much, Eduardo. Thank you for the precise time. So we got a few questions. Does anybody want to pose some question? So I don't see nobody asking here.
I think Albert raised the hand.
Sorry, I cannot see Albert.
So it's amazing work. Congratulations for that. But you showed the last picture about satellite image. Would it be possible for you to fit your framework with real image or a distribution, and how can you parameterize your model to account of that specific scenario that you saw?
Yeah, it's hard to talk about it because it's a hard problem. For instance, for this case of the phytoplankton, we found theoretically that patterns are different of the phytoplankton are different if the predator, the zooplankton, which is around everywhere, is smart or not, or basically if the predator is sensitive or is basically sucking everything object. So we could understand how the predator behaves from the images of phytoplankton. So it's like a very cross inference. We could look at the images, which are easy to obtain, and see what the other guy is doing, which is like very indirect. So we showed this theoretically. This is a recent bioarchive paper, and we actually saw that in data. We managed to show that patterns are different, and not only that the patterns change when you go for different densities. Let's summarize. But it's really hard to, when you are in this interface between physics and like ecology. So like the ocean is a very complicated and very complex. So every time you try to define what is a zooplankton, what is a phytoplankton, an ecologist is going to say, oh, but not all phytoplanktons are like this. Not all. So it's really tricky to create a model, and we had this problem, but we managed to do that. But it was hard to tell a convincing story for an ecologist. So it's an ongoing challenge. Yeah.
So much.
Some other question? I cannot see. I don't see the hands, but may I ask something? So you reach the Turing-like system pattern in one of the limits. In the opposite limit, there is something happening, or?
Yeah. So one side is the Turing, and the other is the non-local.
Non-local.
Yeah.
And do you have quantitatively, which is the separation of scales that you must have to see patterns? And you mentioned a transition also, no?
Yeah.
Critical transition. Can you comment more on this continuous change of rate?
Yeah, that's a good question. We haven't looked this as a like a critical or a transition, in the sense that to look for the critical point or even try to define like this as a transition. But yeah, I don't know, like here you can see we put this dashed vertical line around the 1. So this would be when the population signal are moving in the same rate. So this is will be the divider. So if you go to the right, the signal is becoming faster than the population. So when the population moves or grows or decay, the signals are like diffusing and decaying in between those events of movement and reproduction. So I don't know if there is a critical. Maybe it's like a very slowly growing. So it's hard to define this, but it's it's an interesting question. But yeah.
Have you simulated artificially to see the emergence of patterns, or?
If I simulated what?
The system with the different scale, the different time scale of messaging.
Yeah, it's this. It's here. It's this plot. So this was this. But yeah, we haven't. So yeah, there are many ways to improve the methodology here. You can do like a stochastic PDE style, or you can do agent based, and yeah, agent based I think would be better to see this.
Okay, thank you. Any other question? No? So we will thank Eduardo.
Thank you.
Thank you very much for the presentation. And so we go to the next speaker, Hugo Maia. Hugo, can you connect?
Yes, hi. Can everybody hear me well?
Yes, yes.
Okay, let me just share my screen. One moment. I think can you see it, the presentation?
Okay. So let me briefly present you, Hugo. Hugo Maia is a PhD fellow at the Federal University of Viçosa, Minas Gerais, Brazil. Here is the advisor, Silvio Ferreira, present, I see. And Hugo is going to talk us about contagion dynamics on simplicial complexes and hypergraphs. So.
Okay, so first of all, thank you so much for the opportunity to be here presenting my work to everyone. This work was done in an internship program that I just went to Universidade Zaragoza with Professor Yamir Moreno and Guilherme de Arruda, who is a collaborator of ours now. He's a researcher at Unicamp, and as Professor Celia just said, is about contagion dynamics on higher order networks. We're going to be discussing some differences over the formalism of simplicial complexes and on hypergraphs. So I know that higher order networks are relatively new terms, so forgive me for spending some time discussing what they are, some motivations, their formalisms and everything. The thing is that traditional, what we call pairwise networks, they are limited to representing any type of interaction by a combination of pairwise interactions, right? So if you look here in this network as an example, we have a groups that we can be representing as just an amount of pairwise interactions. This is what we can model, what we can call as groups within a network, right? This on itself wouldn't be a problem. Some systems are in fact linear by nature. The groups are nothing more than linear combinations of pairwise interactions. But what if that's not the case? What if we want to expand our models, our systems, to something that in which a group interaction isn't just a linear combination of, say, for example, in a group, a 3-body interaction here, a combination of 3 pairwise interactions. What I'm trying to say is that networks, or common traditional networks, they are in fact a many-body system, but they do not include on them a many-body interaction, meaning a group interaction that isn't something more than just the linear combination of pairwise or 2-body interactions, right? So, of course, the necessity of using this will depend on the system you're interested in. Some systems, for example, power grid network, the power consumption on this generator that's connecting to these 2 houses is, well, it's nothing more than just the sum of the consumption of the first house plus the second house, so it would be linear by nature. But what about if you want to study social interaction networks? 3 friends go out for dinner and share their opinions, or some new informations and something like that. Is it the same as the 3 of them interacting separately through pairs? I don't think so. And for example, if you take WhatsApp or any other type of social networks, you can have groups, or you can have separate chats between the people inside that will be inside that group, right? So it's something different that we are now starting to look at, see what type of phenomena they can occur from higher order networks. Some few examples, other examples just to motivate us to start studying higher order networks are the first one, cerebral networks, that some works recently pointed to some evidence that neurons activate in groups, something that will be different than them activating separately or pairwisely. Another example, I like this example, it's an example of an ecological network of this monarch butterfly. What happens is this butterfly, its food chain, its interaction with other species, it can be either eaten by this bird or pollinate this flower. I forget their names, but if by chance it pollinates this flower, it acquires a taste that is unpleasant to the bird. So even though the bird doesn't directly interact with the flower, this is a three-body interaction. All interactions depend on the existence of another one of these species. So it isn't just a combination of pairwise interactions. Another good example, and this is the one that interests us the most, is of contagion networks. Okay, so if you think about a virus infection, granted, the direct transmission of one person to the other would be a pairwise transmission. Think of it as 3 infected individuals comes into contact with one susceptible one. The viral load being transmitted to the susceptible one would be just the sum of the other 3. Okay, but what about indirect or aerial transmission? What if people are together in a room? They didn't necessarily talk to each other, but they were in an enclosed environment. So when they stay there for after an amount of time, the transmission occurred and they can get infected through this indirect group transmission in a meeting room, for example. Another example is also contagion network, but for social contagion network. There's this very good experiment, Ash's conformity experiment, that discusses how peer pressure can make people behave in different ways, behave in different ways, or acquire new information through peer pressure, new opinions, which is different than just interacting pairwisely. This is what they call peer pressure, right? So these are some motivations, but let me start now discussing about the formalisms of higher order networks. There are 2 main ways that you can represent a system through higher order networks. Okay, the first one is this one, the simplicial formalism, and the second one is the hypergraph formalism. Both of them are higher order networks. Okay, they are formed up by 4-body interactions, 3-body interactions, 2-body interactions, simplices for simplicial complexes, hyperedges for hypergraphs. They are the same, 4-body interactions, 3-body interactions, so on. But there is this one big difference between both of them. Simplicial complex, it's made out of simplices, and if you have a simplex here, a tetrahedron, meaning a 4-body interaction, for a simplex, you would necessarily have to include all other subfaces as well. So in these 4-body interactions, you must necessarily have 4 3-body interactions, 6 2-body interactions, and yeah, of course, 4 nodes. But hypergraph doesn't have this restriction. So a 3rd order hyperedge doesn't necessarily include 4 3-body interactions, 6 2-body interactions, and so on. So this distinction, it's really important. It's really relevant. There's the key difference between both of them. Some people use simplicials and hypergraph interchangeably, but that's not really proper. And as an example, a co-author network would be a hypergraph because, well, if 4 authors published a paper together, that doesn't mean that all pairs of these 4 authors have published a paper, just the 2 of them, or the same goes for 3 of them, and so goes. So this would be a hypergraph, a system that would necessarily have to be a hypergraph, and wouldn't be very fair to use a simplicial complex formalism on this system. But as I said, we are interested in discussing contagion dynamics. So another thing that we will adapt to higher order networks is the SIS model. I'm going to be using here epidemiological terms, but it isn't necessarily just for simulating the spread of a virus. It could be the spread of information, rumors, some fake news. It's just a matter of picking the model. So for the SIS model, we have these possible processes. An infected agent can become susceptible with a rate alpha, or infected individuals can come into contact with susceptible ones and transmit whatever it is with a rate beta for each type of infection, for each type of order. Maybe it's a 1st order infection, a 2nd order infection, a 3rd order infection, because we are now generalizing it for higher order systems. So if these 2 nodes are infected, for example, they're going to be transmitting the infection or the information through rates beta 1 through these edges, and beta 2 through this hyperedge here, which will be a 3-body interaction. Now, do note that I didn't draw this other big hyperedge here active, being transmitting the infection, because hyperedge in this SIS model is only considered active if all but one of its nodes is infected at a given time. So in order for this hyperedge to be active, we need to have 3 nodes activated, 3 nodes infected, and this one only needs 2. Okay, so we have new extra layers of complexity for contagion dynamics in higher orders. One of them is the hyperedge order heterogeneity. You can have that the bigger group sizes, the more hyperedges that you have in your system, you can generate discontinuous phenomena, hybrid transitions, hysteresis. You can also have, you need to keep in mind, different rates, rate of pairwise infection, of 3-body infections, and so on, which can also generate discontinuity depending on the set of parameters. And also that restriction that I just said, the one that you need all but 1 node active at the same time to activate a hyperedge, can be alleviated, can be reduced to needing only 2 nodes, 1 node, or a percentage of nodes active. So this is all to say that we have many new layers of complexity with many new phenomena that can occur, generating discontinuity, hybrid transitions, and as I said. And of course, with it, there's a problem because we now have to simulate these types of systems. And what often happens is because since we have many new layers of complexity, many times what happens is, well, the algorithms become either imprecise, they can become inefficient, or they can either, well, be used for a very simplified version of the network structure or the model. And we don't want that. We want to be able to simulate complex structures and different types of models, different types of infections, and whatever they be. So we set out to propose an algorithm that will be precise, will be efficient, and will be general for simulating lots of different types of contagion in lots of different types of higher order networks. Since this is a little bit methodological and very specific, I won't delve into many details, but I'll put up a reference in just a few slides of our version of our algorithm. But let me just cite what we did that was so efficient, is that instead of this is the traditional algorithm, this is the one that we are proposing. Instead of just building lists, very complicated lists, with all possible processes, we are now building lists that includes possible processes, but also impossible processes. And these lists are simpler, they're easier to access, they're easier to build, they're easier to update than the proper list with all possible processes. The thing is that now, since we include impossible processes, if perchance we select an impossible process to happen, we don't execute it. We make a phantom process occur, meaning that we count for time passing, but we don't change the state of the system. And this makes the simulation to be exact, to only have possible processes happening. And this key detail that makes the lists much easier to work with made the simulations much more efficient. Let me show just a couple of examples. First of them being a power law hypergraph, and we tested 3 different regimes: a regime with weak higher order effects, moderate higher order effects, strong higher order effects. And the first thing to note is that these black and red curves here for GA and GA+, that they scale, the time it takes to run a simulation on them scales with the system size squared, while our optimization, our algorithm, the complexity scales with the system size linearly or almost linearly, right? Taking this point, for example, of a network, a good-sized network with 32,000 nodes, the simulation time reduces from days or hours to mere minutes, meaning a 99% time reduction with our proposed algorithm. We also tested for some real higher order networks that we acquired from this database on the XGI library, which is a Python library for higher order networks. And what we saw is that our versions of the algorithms, these optimized Gillespie algorithms, or OGAs, for these huge networks, it outperforms all the others more common implementations by a lot. Okay. So, well, as of now, this paper is still on archive, but we do have a repository on GitHub that you can access. We try making it very, very easy to understand, very easy to use. So if this is something that sounds interesting to you, please go take a look. And nothing on this work would have been possible, or at least would have been much more difficult to simulate without this algorithm. So that's why I took a few amounts of minutes to discuss this. But let's move on to the heart of the matter, which is what we are studying. In order to properly analyze what are the effects of either the simplicial formalism or hypergraph formalism, we are going to start discussing what are these nesting coefficients. The name is inspired on this paper here. I put up the reference on top. And what it is, it's a metric of how many mth order hyperedges are contained inside a larger hyperedge H divided by the total number, the maximum number of hyperedges that would fit inside the bigger one. It's easier to understand with this picture. Take this hyperedge of 4 nodes, for example, has these 4 cases. This first one, we have a pairwise nesting coefficient equals to 1 because, well, it could fit 6 pairwise interactions within the nodes from this hyperedge, and there are 6 pairwise interactions within here. This is another example for a nesting coefficient of half, because it has 3 and could have fit 6, and the same here, where it could have had 6, but it has none. All right, so by definition, we are using this because by definition, simplicial complexes are always in this case here on the left, that the nesting coefficient is always maximum, is always equals to 1, while random hypergraphs will have something next to 0. Of course, sometimes they will have some few connections pointing inside, but it is next to 0 nonetheless. Okay, we will focus on a network's overall nesting coefficient. It's just we take an extra average here to measure the average amount of m, lowercase m, hyper edges that are inside uppercase m hyper edges. Is just an average, and let's show this picture here. And we are going to be varying the nesting coefficient in a simple way. We just build a simplicial complex, then we start rewiring nodes from hyperedges. The rewire is very simple. It's done like this. We pick 2 hyperedges of the same size, and then we switch 2 random nodes from it. And as you can see, if we keep switching the nodes from random hyperedges, the total nesting coefficient drops exponentially. And now, this is just a figure to show how it happens. This figure is a figure for the nesting coefficient. For example, this square over here is the nesting coefficient from first order to 4th order, meaning how many pairwise interactions are found inside hyperedges of order 4 or 5 body interactions. And as you can, we keep rewiring the nodes through this parameter f we are using here. It drops to around 0. This is a simplicial, full simplicial complex, the one that we start off with, and then we keep rewiring it, reaching a formalism more similar to the hypergraph formalism. Okay, so how does this impact on contagion dynamics, specifically on the SIS dynamics? Well, it impacts in 2 main ways. Looking at this figure here on the right, the f equals to 0 is the case for the simplicial complex with maximum nesting coefficient, this blue curve. You can see that the transition value, the threshold value here, for the density of infection nodes as a function of the first order spreading rate, it's closer to 0. It's smaller than when we start rewiring the nodes. So going from a simplicial complex to a hypergraph or something, a random hypergraph with little to no nesting increases the threshold value. And also the second way that it changes is that besides increasing the threshold value, it widens the hysteresis loop. So the discontinuity is wider in the hypergraph formalism instead of the simplicial formalism that the transition is, in this case, for this network specifically, it's continuous. This becomes clearer to see when we vary the higher order spreading rate. We define the higher order spreading rate like this. The first order spreading rate is just beta 1, but the spreading rate for trios, quartets, 5 body interactions, and so on, we are defining it as just this lowercase b. And this is our result. This is what we found. This case for the left is for a simplicial complex. This case for the right is a random hypergraph with 0 nesting coefficient. And this case in the middle is, well, something in the middle with some nesting coefficient. We found that there are three regions. First one, with the only stable point being at the density of infected is equal to 0, meaning that there's no outbreak. The region number 3, the only stability, it's with the density of infected bigger than 0, meaning that we always have a disease outbreak. And the region number 2, it's a bistable region, that both of these points have stability, and it's inside the hysteresis loop. And as you can see here, the bistable region, it keeps getting only bigger while the nesting coefficient gets smaller. So in a way, we can say that for simplicial complexes, the outbreak is easier, but for random hypergraphs, the outbreak is more expressive, okay? So we decided to measure the nesting coefficient for some real higher order networks. What we found is something like this. These are the same networks for the database I mentioned a few slides ago, and we found that some networks have bigger nesting coefficients, other ones have smaller nesting coefficients, but something curious is that the nesting coefficient is most often than not concentrated within smaller orders. As you can see here, there is little to no nesting at higher orders because, well, you can think of it as like, it doesn't make much sense to have a group of 10 individuals and then other groups of 9 or 8 of the same individuals. It's a little bit of a redundancy. These are the nesting coefficients for a few of the networks that we measured. And, well, the presence of these nesting coefficients isn't a matter of structural restrictions or anything like that. It is something that happens. We know that because we rewired these networks keeping the same distributions, and we saw that the nesting coefficient of a null version of this network, a randomized version of this network, drops substantially, okay? Then we set out to test our hypothesis that would correlate the hysteresis length with the nesting coefficient. We did this in a little bit of a peculiar way. We took note of a few points. This is the same figure as before for the synthetic higher order network that we used, but it's just to exemplify how we define the hysteresis length. We took 2 points. The first one being this one, the threshold value, when there is no higher order terms, there is no higher order infections, no higher order rate. And then we took a note of another point that is the threshold value, the pairwise threshold value, when there are a specific amount of higher order rates, right? So when we divide these two values, we have something that we call the length of the hysteresis loop. It's our measure for the length of the hysteresis loop. This precise point that we take here for B0 to measure the second value for the threshold, it's exactly the point that this lower branch here reaches 0, meaning that when the transition value for the decay dynamics, because it's bistable, it's equal to 0 with this parameter for the higher order spreading rate. And this is what we found. These are all the higher order networks that we measured, and this axis we have the hysteresis length, and this other one we have the average nesting coefficient. And there is some sort of correlation or inverse correlation between the nesting coefficient and the hysteresis length. We measured the Pearson coefficient and the Spearman correlation, and they do indicate very negative values, showing us that there is really an inverse linear relation, a very confident inverse linear relation between nesting coefficient and the hysteresis length for several different types of networks. Okay, we were also interested on seeing how different nesting patterns impact on the dynamics. As I said, this pattern here with nesting more concentrated on smaller orders is the one that we find in real hypernetworks, real higher order networks. We also tested different ones with more balanced nesting patterns and then one more focused to higher orders. And curiously enough, the way that it is in real life networks is the one that enhances the most contagion dynamics. As you can see here, the curve for the pink case, the one with the real life nesting, it's stronger. The contagion dynamics, it's stronger. I find this very interesting, actually. Coincidental or maybe not. The way that real networks organize themselves is the way that enhances the contagion dynamics. So just to finish off with a thought, if you put a line in entropy, the the simplicial complex will be on one side, and the random hypergraph will be on the other side. Why? Because, well, simplicial complexes are like crystalline structure. You have many information about which way some hyperedges are pointing, because you know that they're always going to point inside other hyperedges for simplicial complexes, while for random hypergraphs, it's more like a gaseous structure. It's very random, it's very unorganized. And somewhere on the middle, we have our real higher order networks. So this got us thinking about the concept of the edge of chaos, that complexity happens most between order, as the simplicial, and chaos, as in random hypergraphs. So it's a little philosophical and very interesting, because as I showed you in a few slides back, the scales and patterns of mixed nesting coefficient, as in real hyperorder networks, it's more complex, has more interesting things to be looking at instead of just simplicial or just random hypergraphs that are somewhat simpler. So as some other concluding remarks, I like this phrase from this paper that discusses that real systems have higher order effects. Not all of them, but lots of them have higher order effects. So it makes sense for us to start thinking about higher order networks as a way of modeling, as a way of studying these systems. We propose this algorithm that is very useful for simulating contagion dynamics on higher orders. We hope it will be useful for the community. And with it, we were able to discuss the effects of nesting coefficient, how it alters contagion dynamics. We tested it for some empirical networks, some of them very, very big, some of them smaller. And we also tested for different patterns of nesting coefficient. And we saw that it is really a relevant aspect, a relevant metric that should be taken into account when studying higher order systems. I guess this is it. Thank you so much for your attention. Thank you for the organization, for the funding agencies. And if you have any questions, feel free to ask them or contact me with my email over there.
Thank you very much, Hugo. Do you see some hand raising I can see for questions? Well, I cannot see. Sorry, but do you see Hugo?
Yeah, there's a question by Eric Rosen here. If I can read it, it's just if I understand correctly, a mean field SIS model could include higher order interactions by including terms on the form of SGI times elevated to M. Yes. Have you tried anything analyzing models like that? For example, do you find if you can analytically find the new threshold values? Yes, that's exactly it. The model for the mean field equations, let me just return to this. I don't have any slides, but it's just return to the slides here. As Eric asked, the mean field equations are exactly like that. We have some higher order terms with the density of infected nodes. There are several different papers that discuss mean field theories. We didn't look at mean field theories here for this case, but with only the mean fields, you can return something similar to these figures over here. No, this one is good enough. These figures over here that includes discontinuity, and we can find critical values for beta 1, we can find critical values for beta 2 and others with mean field equations that will be with higher order terms. We didn't do it here, but yes, it's something that you can most definitely do. Thank you for the question. Let me return to the...
I will ask you something.
Yeah.
Have you considered, or planning to consider other dynamics, for instance, social opinion dynamics or dynamics with other different features? I mean, what you expect to be impacted, no? But this natural strategy.
Yes. We do plan on it. There are lots of other models or lots of other types of dynamics like synchronization, like opinion dynamics that are being adapted to higher order networks. I myself haven't worked with opinion dynamics on higher orders or synchronization dynamics on higher orders, but we are looking at other types of compartmental models. For example, here we studied mostly the SIS model, as I show it here, right? For susceptible, infected, susceptible model, but we are also studying SIR models, SIRS models, a version of the rumor model for higher order networks and what it impacts. Some things are impacted in a way similar to the SIS, but some others are not. It's really model dependent, and there are lots of new interesting avenues there to be studied. So yes, that's something we are planning on seeing.
Okay, thank you very much.
Thank you, thank you.
I don't see other questions. I think there are no more questions. So thank you very much, Hugo. And let's thank the speaker. So thank you very much.
Okay, I think that's good. Please come up with the program.
Hilda?
Yeah, we have